Hello, dear students from a well recognized international university with a background in basic molecular biology, immunology, plant science, and uh, cellular signaling. So, in the next 5 to 10 minutes, I'm going to talk with you about my research interest, what I've done the last couple of years. So, what my research is all about is it's about uh, the application of basic science in plant defense engineering. And I was working on rice the last couple of years. And so rice in some parts of the world gets attacked by this bacterial pathogen, which is called Xanthomonas oryzae. And so this infects the leaves, it multiplies inside the leaves, and then the two principal defense outcomes. One is like the plants can be susceptible, so the plant actually can't recognize the bacteria, it's kind of blind to it, and the bacteria happily multiply within the plant cell, take up all the energy, use the energy of the plant, and the plant gets sick and wilts and dies at the end. On the other hand, on the other side of the spectrum are resistant plants, and so resistant plants have receptors or antennas. In this case, we studied X antenna, which is called XA21, and this antenna recognized a signature of the bacteria, which is called RUXX. So when XA21 recognizes signatures, this protein of the bacteria, um, it can actually initiate a defense response and fight off the bacteria, stop the replication of the bacteria, the multiplication of the bacteria, and quarantine, so to speak, the bacteria, prevent it from multiplying, and the plant stays healthy. So, as you all know, plants are really important for all of us. They produce most of the oxygen we breathe. You all probably had breakfast this morning. Maybe you had a toast. Maybe you had some cornflakes. Maybe you had some rice. So it's really obvious that the plants are really important. But plants don't grow in isolation. Plants are constantly surrounded by microbes. And these microbes could be viruses, oomycetes, fungi, or bacteria. And because plants are so energy rich, they have a lot of like starch and sugars. Also, not only we want to eat them, but also like these bacteria, fungi, viruses, and oomycetes want to get the energy out of the plants. So the plants need to be able to defend themselves, similar like to all other organisms. Plants also have an immune systems, and that's what I'm gonna explain to you in the next slide. And the take-home message really is here is that you have like two layers of immune response. One is happening in the apoplast, so at the outside of the cell, and one is like intracellular. Let's focus first on the extracellular part. So fungi and bacteria and oomycetes, they secrete molecules or release molecules into the extracellular space, into the apoplast, and plants have these receptors which stick out into the apoplast to recognize these molecules. So one receptor recognizes one molecule at a time, and after this molecule is recognized, the receptor relates the signal into the inside of the plant cell, into the cytoplasm, and often these receptors are coupled with an intracellular kinase. And this again is coupled with a co-receptor, so when the main receptor recognizes the molecule, it initiates signaling cascade via phosphorylation at the kinase, and this at the end leads to defense signaling, and this can be either major defense signaling, so the plants are fully immune, or a minor disease resistance, as it's called in agriculture. They only have like a small contribution to be not fully susceptible, but only half susceptible. So, and then the second layer of the immune system is really the recognition of molecules inside the plant cell by so-called nuclear binding site leucine-rich repeat domain containing proteins. Again, these are receptors recognize intracellular molecules from the pathogens, and This recognition event, in a similar manner, can either be a major one, but the plants are totally immune, or it can only, on the other side, also just contribute to a partial resistance, like those plants are not fully immune, but can contain the pathogen. So in the absence, really, of these immune receptors to recognize these so-called effectors, which are mostly small proteins, these effectors are used by the pathogen to generate favorable conditions in the plant for the pathogen for nutrition, suppression of immunity, and so on. So these two layers from intracellular and recognition, extracellular recognition, that's kind of the take-home message. And I study mostly the part of the extracellular recognition events. So how do you study this? So when these receptors recognize molecules from the bacteria, in this case, a plethora of signaling events is triggered. For example, one is like a calcium influx across the membrane. Then you have the generation of reactive oxygen species like hydrogen peroxide, zinc oxygen. So these are high energy oxygen molecules which easily react with other macromolecules inside the cell. And on the other side, you also have like kinases, which are an integral part of many of these receptors, and these phosphorylate other molecules, which phosphorylate other molecules, which leads to this kinase cascade. And at the end, this whole response accumulates into the reprogramming of 5 to 10% of the genome and the upregulation of many defense genes. And this leads to defense response and to the disease outcome at the end. Either the plant is susceptible or is resistant, as I mentioned initially. So what do we do? How do we measure this? So what we can measure is the reactive oxygen species by a light-sensitive camera, an enzymatic assay, and as I mentioned before, I was working on rice. So if you have a rice, in this case we call it Kitaki. It's a rice variety of one of the over 1,000 rice varieties, which doesn't have the immune receptor to this corresponding molecule, which we call RUXX. So you put this RUXX on, nothing happens. But if you have the immune receptor and the right form of RUXX present, then you get an immune response within like 30 minutes, which peaks around 1.5 hours. So and you can measure this really easily. And another defense outcome we can measure, again, is defense gene expression by quantitative RT-PCR, like reverse transcription. So in this, we measure the expression of genes which we know are related to the defense outcome. And one of the very classical markers is plants is pathogen-related proteins, PR, TMB in this case. And again, a rice variety which doesn't have the receptors, like blind, and the rice variety which has the receptor actually recognizes the molecule if you just put it on it by reprogramming of its genome. So, and this leads to defense outcome. So here you have show you a plant, rice plant, which is infected with the bacteria which has this RUXX molecule, and the plant has a immune receptor, so you see very little disease progression, so it leaves it really green, so the plant is healthy. However, if you infect this plant with bacteria which lacks this molecule, which is now recognized by XA21, you're like fully susceptible. You see like this long lesions and like this yellowing, so the plant clearly suffers. And instead of like just taking a picture, you can also like quantify it, but just by taking a ruler and measuring this. So if a plant is resistant, the disease progression is very minor, only a couple of centimeters. But if you're infected with a bacteria where RUXX is like mutated, then you get these really long lesions, like 15 centimeters. And then what you often have to do with genetics is to show that your mutant you made is really causative for your phenotype is you put back the gene which you mutated in its native form, and then you reverse the phenotype, and that's what we showed as well. So you have a mutant of the bacteria which expresses the wild type protein again, and this is then not recognized, this is then recognized and the plant is resistant. So with these different techniques and something like more techniques which are not going to go into detail, we could show first that plants which have the immune receptor can recognize this molecule in the absence of the bacteria. So yes, this is probably the molecule which is recognized, and also this is probably also the only molecule from this bacteria which is recognized by XA21 with this specific immune receptor, because if you take away this molecule from the bacteria, plants that have the immune receptor become susceptible because they can't recognize the bacteria. And that's what's summarized in the slide here. So it's work of 4 years, you have to mention, with like lots of hands involved. So if you have rice plants which are infected by the bacteria which express and secretes this molecule RAXX, this then can be recognized by XA21 via a molecular recognition event, and this leads to immune response and the plant will look healthy and are resistant. On the other hand, if these bacteria infect plants which lack this immune receptor, RAXX is not recognized and the plant is susceptible. And so now we can use this knowledge to monitor bacteria in the field for having this molecule for RAXX, and we can plant rice varieties which have this immune receptor and they're going to be resistant. So, and this is important in the field, as illustrated by this picture. So here in the background there's a rice variety which is like resistant to the bacteria because it recognizes a certain molecule. So the disease control is genetically encoded in rice, in the genome. In the foreground, you have like a different rice variety which does not recognize this bacteria, Xanthomonas, and you see the blight, total devastation of this field, and of course you lose all your grain. So it's really important to study this molecular signal event between plants and pathogens to be able to have these green fields and make an informed decision what kind of plants you should grow to contain pathogen spread without the application of too much pesticides. Thank you.